Next speaker is Dr. Tiffany Sills. Tiffany is currently at Baylor College of Medicine. The title of her talk is Characterization and Molecular Regulation of Hemogenic Endothelium within Aorta-Gonad-Mesonephros for the AGM region in the Midgestation Murine embryo.
Good morning. My name is Tiffany Sills. I'm at Baylor College of Medicine. I'm an MD PhD candidate. And as you can see, the first title is very long, so I decided to shorten it a little bit. So what I'm talking about today basically is parallel development of endothelium and blood, specifically hematopoiesis in the AGM region. And so hematopoiesis occurs in two waves during urine embryogenesis. There's a primitive wave, which is primarily erythrocytes, and there's definitive, which by definition forms all cells of the blood lineage, both myeloid and lymphoid, and these cells are capable of engrafting into an irradiated recipient. And so this occurs in many different locations. So it begins at E, or embryonic day 8.25 in the yolk sac. It then moves to the AGM region at E10.5. These cells then seed the fetal liver at 14.5, and then finally the bone marrow will be their residence from 18.5 throughout the rest of their lives. And so as I said, I'm focusing on the AGM region. So the reason why my lab would care about hematopoiesis, because we're primarily a vascular lab, is because we believe that the endothelium provides the origins of hematopoietic stem cells during embryogenesis. And so this model kind of represents what we believe is going on. So to orient you, this would be kind of dorsal ventral, and this would represent a large blood vessel such as the aorta. And what we have initially is a primitive endothelial cell, which then becomes specified to become a hemogenic endothelial cell. These then give rise to hematopoietic progenitors and or hematopoietic stem cells, which finally give rise to blood. So by definition, hemogenic endothelium would be a specialized type of endothelium that has the potential to differentiate into HSC and or hematopoietic progenitors. And so to give you an idea what this would look like, here I have a frozen section from 11.5 in the AGM, and I've stained for VE-cadherin, which is an endothelial cell marker, and c-KIT, which is a stem cell marker. And what I'd like to draw your attention to are the areas of co-expression within the vessel wall of these markers and the interesting morphology of those cells. And so this will loop. And so what you can see is, especially looking down here towards this side of the vasculature, you can see that there are cells with kind of a bulging morphology into the vessel wall, and the red cells that you see on the inside are obviously blood cells. And so according to our model, these cells that, or these areas are areas of hematopoietic activity where blood cells or blood progenitors are being formed and then released into the lumen. And so then to kind of bring this back to a larger scale model, what we're talking about is essentially differentiation of specialized forms of endothelium. So as I said before, initially we start with primitive endothelium, and the markers for that will be markers like CD31 or PCAM, FLIC1, VE-cadherin, and CD133. Then through a variety of both extrinsic and intrinsic factors, this is specified into arterial endothelial cells, venous endothelial cells, and hemogenic endothelial cells. And so work has been done primarily in the yolk sac thus far to characterize hemogenic endothelium. And so these are some of the markers I'm going to tell you about now. So the first thing is it actually has the SP or side population phenotype, which I'll review in the very next slide. Positive for markers like CD31, FLIC, negative for CD45, which is a marker of definitive blood cells, positive for stem cell markers such as SLAM or CD150 and c-KIT, and also positive for hematopoietic transcription factors such as GABA1, tau, SCL, and RUNX. And so as I said, I'd briefly go over the SP or the side population. So Kenny was talking about one way that you can isolate hematopoietic stem cells using cell surface markers. However, there are a few other ways you can do it. So one way you can do is use an experiment where you actually stain all the cells with this DNA dye called a Hoechst dye, and then you look at the rate of efflux of that dye from the cells. And so what happens is, initially this was used to isolate HSC from adult bone marrow, and it seems that the stem and progenitor cells within the total population have the ability to efflux this dye at a rate much quicker than the rest of the cells in the population. So what you can see in this profile, essentially is the majority of your cells kind of lie towards the bottom of the profile and off toward the right, and then you have this small but distinct population that comes off the side, hence the name side population. And, so initially, like I said, it was used to isolate HSC. It's since been shown to mark numerous stem and progenitor cells in different tissue types. And what's unique about this is it actually represents the functional property of the cells, as opposed to just cell surface markers. And so the first question we had was actually, can we isolate an SP in the AGM during the initiation of hematopoiesis? And in fact, we were able to do so. So beginning at E9.5 and going out through E11.5 in the AGM region, we're able to reproducibly isolate an SP population, and that frequency clearly increases over time. So next we wanted to know if this population was enriched for hematopoietic activity. So we used a blood colony forming assay called a Methicol assay to do that. And so here what this chart represents is we took the AGM region, we microdissected it out from mouse embryos at these different time points, so 9.5, 10.5, 11.5, and 12.5, and we fractionated that into SP fraction and non-SP fraction of cells. Then we plated 1,000 of each type of population in the Methicol assay, and then we looked for blood colony formation. And so we found the following. So 9.5 yolk sac just represents kind of a positive control for me to be sure that the assay is working correctly. And here you can see in this legend quickly that the different colors represent different colony types. So this kind of light blue or lilac color would be an erythroid colony. The purple is GM, which is a granulocyte and macrophage colony. And the beige or the yellow here is a multilineage colony having many different cell types. And what you can see is for every time point that we tested, the hematopoietic activity was restricted to the SP fraction as opposed to the non-SP fraction. So then we wanted to kind of look at the SP fraction in more depth and kind of understand something about the cell surface markers expression in this population. Because we know that traditionally, the SP is a heterogeneous population of cells. And so typically you use the SP and then you can also use a combination of markers to then kind of pinpoint the exact population you're interested in, or most interested in. And so here kind of I started out by using a lot of the same markers that have been isolated in the yolk sac to see if these hold true in the AGM. And so what I found is in the SP fraction, actually the majority of the cells are negative for FLIC1, which is the endothelial marker, and they're also negative for SCA1, which is a marker that's used in the adult system to help differentiate the SP for hematopoietic stem cells. Then when looking at the stem marker c-KIT, we see that it's about 50-50 in terms of expression. And then looking at CD31, which is primarily an endothelial marker, but it marks a few other cell types from the mesoderm, we also see that it's relatively split between positive and negative. But the CD31 fraction actually separates pretty nicely into a high and low fraction. And so here you can see that relatively is equal as well. And here, so then we took 100 cells from each of these different populations and put them back in that Methicol assay to see if we could kind of further narrow down which population within the SP is responsible for hematopoietic activity. And so these red arrows here, represent which fractions grew colonies in our assay. And so what you can see, interestingly, is that we actually had colony formation in both the FLIC positive and the FLIC negative fraction. So that's an endothelial marker and then the non-endothelial by expression, excuse me. And then in the c-KIT marker, we actually saw that colony formation was restricted to the c-KIT positive fraction. For CD31, we saw that it was also restricted to the CD31 positive fraction. And then looking at the breakdown of high and low, it's actually in the low fraction of CD31 positive cells. And then finally, looking at SCA, we saw that blood formation actually occurred in the SCA negative fraction, which is actually interesting because it's very different than what you would see in the adult system. And so I just kind of wanted to also move on and just show one other example of some of the imaging work I've been doing in the lab. So one of the things that in our particular field has been lacking is actually in vivo evidence of this phenomenon occurring. There's lots of in vitro data. There's also, interesting staining that people have shown in either whole mount or slides, but we've yet to see this occur definitively. So we wanted to address that. And so essentially what I've created is an explant culture system using confocal time-lapse imaging that would be able to track and understand the behavior of labeled cells. And so here I was actually just using a FLIC1 EYFP transgenic mouse model to set up the system. And just to orient you into what you're going to see, actually here you have the anterior, posterior, or rostral-caudal axis kind of going along this way, and then the dorsal ventral axis, so from back to front going this way. And so here, AL represents the aorta, and then NT represents the neural tube, and everything that you see pseudo-colored in green would be the FLIC positive cells. So when I begin the loop, sorry, you can see the movement of these cells kind of along both the length of the aorta and across the width of the aorta. And you also see some cells that actually migrate out towards the neural tube. So this allows us to kind of understand a little bit more about endothelial cell behavior. And we're hoping that when we find the right combination of markers, we can actually use this, as I said before, to kind of definitively show this process occurring in development. And so in conclusion, what I've shown you today is, one, a subset of endothelial cells with distinct morphologies in the AGM region co-express B, cadherin, and c-KIT. Two, in the Murine AGM, the proportion of SP cells increases over time, but that hematopoietic activity appears to actually decline. Three, in the 10.5 AGM SP fraction, a majority of the cells are negative for FLIC1 and SCA1, while c-KIT and CD31 are expressed on about half of those cells. Four, preliminary data shows that the colony-forming activity within the AGM SP fraction is restricted to the c-KIT positive, CD31 positive, SCA negative fraction, although we see colony formation in both the FLIC positive and the FLIC negative populations. And then finally, that in our lab we've developed this explant culture system for dynamic imaging, which allows us to track the developing endothelium and understand something more about its behavior. And with that, I'd just like to acknowledge my lab. My boss is Karen Hershey, as I said, in the Hershey lab, some of my collaborators at Baylor, the MSTP program, my funding, which is a predoctoral fellowship from the NIH. I'd like to thank the Meyerhoff Scholarship Program for the opportunity to speak, and I'll take any questions.
So in addition to the confocal or the microscopy, can you transport the cells from one embryo to another, or your transgenic marker to see that there's an application? Is that possible? I don't know.
So it is possible. So one issue I think in embryonic HSC is the issue of when are these cells actually mature enough to engraft. And so obviously in the adult system you can take them and transplant. That's not a problem. There have been some issues with that with the system, but I'm hoping that the population I've isolated in particular is actually going to be robust enough to show transplantation and engraftment. So yeah, that's something that you can do.
How do you explain the fact that these FLIC plus cells show colony forming potential?
Right. So I guess by our model, these would actually be a specialized form of endothelial cells that are actually giving rise to blood.
Okay. So do you think that they're free so that those cells eventually will become FLIC minus, or do you think they go directly from endothelial cells to blood-forming cells?
So probably the progression will be something like this. So initially you have a cell that will display the endothelial FLIC, and then as they give rise to these immediate progenitors, then maybe they would lose the FLIC and go on from there. So that's why I think you would have colony formation in FLIC positive and FLIC negative population.
Okay. So maybe you're looking at a cell type in transition.
Yeah.
Perhaps. Any other questions? Great. Thanks, Tiffany.